Hello and welcome to another episode of Science on the Edge. I'm Dr. Mark Darcy, and in today's episode, I'm going to be talking about the effects of the extracellular matrix on cancer, on both propensity towards developing cancer and on the spread of cancer once it has developed. You'll find out pretty soon that the extracellular matrix is not just a sort of membrane that surrounds tissue and attaches to tissue. It has a very important function in regulating how cells behave, including cancer cells. Okay, so let's get started. By the way, for anyone who would like to follow me on Twitter, my Twitter handle is @MarkOfTheD, @MarkOfTheD. Okay, so here we go, cancer and the ECM. So first of all, the extracellular matrix, ECM, is a very dynamic structure. Its components are constantly being synthesized, deposited, degraded, and remodeled. This remodeling is tightly regulated by a variety of enzymes and influenced by mechanical cues. In cancer, the genetic and epigenetic regulation of these processes becomes dysregulated, leading to an ECM that not only allows, but also actively promotes tumor growth and metastasis. The interplay between the ECM and cancer cells is thus not only physical, but deeply rooted in the molecular genetics that govern cellular function. So the extracellular matrix itself is a three-dimensional network, and it's composed of proteins, glycoproteins, so proteins with sugars, and polysaccharides, which provide structural and biochemical support to surrounding cells. Its roles in cancer are multifaceted and critical to various stages of cancer progression, from growth to invasion, right through to metastasis, even angiogenesis, the formation of blood vessels by the cancer, all influenced by the ECM. Alterations in the ECM can lead to changes in cell adhesion, motility, meaning movement, and proliferation, all key aspects of tumor development. For instance, the downregulation of ECM components, such as collagen, can decrease tissue integrity, allowing cancerous cells to invade neighboring tissues. Should mention here that collagen is something that naturally decreases in its thickness throughout our body as we get older. The collagen, which is in our skin, our muscles, organs and tissues, all goes down in terms of its thickness as we age. So it reduces in its thickness, and the collagen we do have becomes glycosylated, meaning sugar ends up stuck to it, which again affects its behavior and its structure and its rigidity too, which can all have a negative influence on cancer. One of the many reasons why cancer tends to be a bit worse in people that are older than it is in people that are younger. The ECM is involved in the regulation of the cell cycle, and aberrations in ECM components can disrupt normal cell cycle control, potentially leading to unchecked cell division and tumor growth. Cancer cells can induce the local ECM to undergo a desmoplastic reaction, so a stiffening of a tissue that is often associated with the invasive properties of malignant tumors. The ECM influences angiogenesis, so a formation of blood vessels, which the cancer needs to grow past a certain size. Should mention maybe that the center of a tumor, once it gets past a certain size, becomes hypoxic, starts to die, because the cells in the center don't have a blood supply and run out of oxygen and nutrients. So angiogenesis is necessary for a tumor to get past a certain size. It needs a blood supply. It needs to release factors or influence release of factors that will allow blood vessels to grow through it and allow it to absorb more oxygen and nutrients and grow. So the ECM influences angiogenesis, which is essential for tumor growth by supplying nutrients and oxygen. Cancer cells can modify the ECM to create a pro-angiogenic environment. Components of the ECM combine to growth factors, modulating their availability and activity. Dysregulation of such interactions can lead to the sustained growth signaling that is characteristic of cancer. VCM can impact the immune response to cancer too. It may either present barriers to immune cell infiltration or promote a microenvironment that suppresses immune surveillance, aiding in immune evasion by tumor cells. Basically, we get cancer regularly, all of us, throughout our life, or at least the beginnings of cancer. But these early cancer cells are usually just removed by our immune cells. Our immune cells patrol our body and look for aberrant cells and destroy them by a number of processes, but one of the key ones is known as the extrinsic pathway of apoptosis. Now, I've actually written a paper on apoptosis, a couple of papers on apoptosis, so I know a little bit about this. And dysregulation in the extrinsic pathway by a weakened immune system or a dysregulated immune system helps cancer to both develop and spread. Metalloproteinases, MMPs. These are ECM remodeling enzymes that are often upregulated in cancer. MMP activity can facilitate metastasis, the spread of cancer to other tissues, by degrading ECM components, paving the way for tumor cells to disseminate and spread, first to invade, then metastasize to other tissues. The ECM plays a role in the creation of 3 metastatic niches, which are primed environments in distant tissues that favor the survival and growth of metastatic cancer cells. The stiffness and topography of the ECM can influence the mechanical signals that cells perceive, which can affect gene expression and behavior of cancer cells, potentially leading to more aggressive phenotypes. Cancer-associated fibroblasts, CAFs, are also important modulators of ECM in tumors. They secrete ECM components and remodeling enzymes, which can aid in tumor progression and resistance to therapies. Now, targeting ECM and its related pathways has been proposed as a therapeutic strategy in cancer treatment. For example, inhibitors of MMPs have been explored to prevent metastasis, though with variable success so far in clinical trials. Research into ECM's role in cancer is ongoing, however, with a particular focus on understanding how ECM remodeling contributes to the tumor microenvironment and identifying new therapeutic targets within ECM-related pathways. Ironically, if that's the right word, treating cancer can actually make cancer more likely to occur and spread. Basically, if you, for example, have breast cancer and it's treated, this usually involves surgery, radiotherapy, and chemotherapy, all of which is going to massively damage and disrupt the ECM around where the initial tumor was, which makes it more likely that a new tumor will form because its neighboring tissue now has a disordered ECM which never quite recovers from something as severe as surgery and radiotherapy. At the genetic and molecular biology level, the extracellular matrix plays a crucial role in regulating cellular behavior and gene expression, which can have profound implications for cancer initiation and progression. The ECM is composed of an intricate network of proteins and polysaccharides, as I mentioned earlier, that are produced and maintained by the cells within the tissue. These components include fibrous proteins such as collagen and elastin, glycoproteins like fibronectin, and laminins and proteoglycans. The ECM provides structural support to cells, but also acts as a reservoir for growth factors and cytokines that can influence cell behavior. Think about the ECM as a sort of sponge holding on to pro-growth factors. If that sponge gets damaged in some way and these pro-growth factors are released in too large an amount, they can stimulate cells to go into mitosis, to start dividing uncontrollably, which is basically what cancer is. Integrins are transmembrane receptors that mediate cell ECM adhesion. Upon binding to the ECM components, integrins cluster and initiate intracellular signaling cascades. These signaling pathways can affect gene expression by activating transcription factors such as NF-kappa beta and AP1, which regulate genes involved in cell proliferation, survival, and migration. These processes are often hijacked by cancer cells. I'll mention one of those, NF-kappa beta. I'll delve into a bit more detail. This is a pro-inflammatory cytokine. Stimulates the immune system, stimulates inflammation. Excessive inflammation is a cause of cancer. It is a cause of cancer. Inflamed tissues are more prone to cancer. An example of this would be people that are very overweight, carrying a lot of subcutaneous fat. Well, that very fatty environment actually activates the immune system and inflammation. It's a pro-inflammatory environment, which is one of the reasons why people that are very overweight are more prone to certain types of cancers than people that are carrying less weight. Matrix metalloproteinases. So these are enzymes that degrade ECM components and are crucial for ECM remodeling. They're encoded by a family of genes that can be upregulated in response to various signals, including those from cancer cells. By degrading the ECM, MMPs release sequestered growth factors, mentioned earlier, that can then activate receptor tyrosine kinases on cancer cells, promoting proliferation and survival signals. There are tissue inhibitors of metalloproteinases, though, TIMPs. These TIMPs are natural inhibitors of the MMPs and play a role in maintaining ECM homeostasis. Alterations in the balance between MMPs and TIMPs can lead to excessive ECM degradation, facilitating cancer invasion and metastasis. ECM stiffness and mechanotransduction. Changes in ECM stiffness can alter the mechanical forces experienced by cells, which in turn can modulate cellular signaling and gene expression through a process called mechanotransduction. Cells sense the mechanical properties of their environment through mechanosensitive proteins, such as the previously mentioned integrins and other molecules like YAP and TAZ, which are involved in what we call the hippo signaling pathway. This can lead to changes in gene expression that favor oncogenic transformation and cancer progression. Interestingly, however, when it comes to someone that's already got cancer, the spread of the cancer to other tissues can be reduced by physical activity, at least to a level. Basically, any cancer cells which make it into the bloodstream, they can be affected by blood pressure. What do I mean by that? I mean that when you exercise vigorously, blood pressure goes up by quite a bit, the force of the blood changes quite a lot, and this puts a shear force on cells in the bloodstream. And if there happen to be any cancer cells in the bloodstream, this shear force can actually trigger them to die. They want to die anyway, cancer cells, they just can't. But these shear forces, by intense exercise, can actually kill cells. It's been shown in a number of studies that people that do exercise vigorously following a diagnosis of cancer, the cancer tends to progress on average more slowly, and recovery rate is higher, and metastasis tends to be reduced, or at least the levels reduced, at least in some studies. Epigenetics. The ECM can influence the epigenetic state of cells. Epigenetics just means which genes are switched on or off in a cell and by what degree. For example, changes in the mechanical properties of the ECM can affect what we call histone acetylation and methylation. These are mechanisms that switch genes on and off, leading to alterations in chromatin structure and gene expression. So basically, any kind of mechanical force on the body will change which genes are switched on and off in different cells. Hypoxia, so low oxygen levels. Tumor hypoxia or low oxygen conditions can lead to ECM remodeling. Remember, it's usually hypoxic in the center of a tumor. Hypoxia-inducible factors, HIFs, which are stabilized under low oxygen conditions, can induce the expression of genes that alter the ECM, such as those encoding for collagen and MMPs. These changes can create a microenvironment that supports angiogenesis and invasive behavior of cancer cells. Okay, that was a brief overview of how the extracellular matrix influences cancer growth and development. If you want to know a little bit more about what I've got to say about anything, then you can follow me on Twitter at @MarkofD. There will be more lectures coming up on my channel about cancer. It's such a big deal. It affects about 50% of all of us at some point in our life. Well, about 50% of us will get it. We're all affected in some ways. We will know someone that gets it. So I will be making more, well, putting together more talks about cancer and its treatments in the future. But that's all for now. And I look forward to speaking to you all again very soon. Goodbye for now.